and welcome to our short discussion today on high-throughput X-ray footprinting with the team from Lawrence Berkeley National Lab. My name is İlker Şen. I am Customer Success Scientist at Proteinmetrics. With me today are Corie Ralston, Facility Director at the Molecular Foundry at Berkeley Lab, Sayan Gupta, Research Scientist, and Lina Kristensen, Associate Scientist in the same lab. So welcome everyone, and thank you very much for joining us. I'd like to start today's discussion with Corie, who's known for helping develop X-ray footprinting as a structural investigation technique for biopolymers. So Corie, there are several types of oxidative labeling techniques available out there. How does your approach improve upon them or complement them?
All right, well, great question. So first off, I wanted to say that all methods have advantages and disadvantages. So I feel like our method is highly complementary to other structural biology methods. And really these days you need to use a suite of different methods if you really want to answer the scientific question that you're asking. But in terms of what we feel are the advantages of our method over other oxidative labeling methods. So first off, we use hydroxyl radicals for the labeling, and hydroxyl radicals have a couple advantages over, say, using proteases. So they're small and they're highly reactive. So the fact that they're small means that you can get really precise structural information. So for instance, you can see labeling of just a couple of residues that happen as a process of something binding to a protein or a protein folding. And the other advantage is them being really reactive means that you can get labeling in a really short amount of time. So you can label, have the event be over and done with, and minimize all the secondary types of reactions that are happening. So the other component of that is we use X-rays to generate hydroxyl radicals, and there's a couple advantages to using X-rays. First off, you can control the dose pretty well with X-rays, so that can be precisely controlled to control the labeling amount. You don't need to add any reagents such as iron EDTA or hydrogen peroxide. You can use a range of buffers. We are limited in our buffers we can use, but you can use, you can generally use buffers that proteins are happy in. And I think one of the big advantages is that it's not a science thing, but it's the fact that we're at a national user facility means that we can have a user program. So this isn't like an experiment we're doing in our own lab and you have to be friends with us to do the experiment. You know, you can actually apply for time on the footprinting beamline, and we can help you with your project. So I think that's a big advantage. In terms of disadvantages of our method, so one of the things that's an advantage is also a disadvantage, the fact that it's at a synchrotron. Synchrotrons aren't up year round. You do have to send in a user proposal to get beam time. Other disadvantages, so we're not super high throughput, although we're working on that. So it takes a while to do a footprinting experiment and get results. It's not like HDX, which I understand you can buy a system that'll just do that, and companies have HDX systems and they use them, you know, get results immediately.
Immediately is a is a stretch, but yes.
Yeah. Well, yeah, I guess it's all relative. And I guess one of the other disadvantages is you do need mass spec to do the method, and that can be complicated, and the analysis can be really complicated. So.
Yeah, that all makes sense. So maybe I'll switch to Sayan for the next one. So for the jet system that you developed, how concentrated does the samples actually need to be? And what buffer conditions do the users need to keep in mind when they're setting up these experiments?
Thanks for this question. So I want to answer this question in two parts. First is the concentration. So we use dilute solution of proteins like 10 micromolar, 2 micromolar to 10 micromolar in phosphate buffer. So this is a standard concentration, and we can run jet. We don't want to make concentration too viscous so that the jet will not work properly. But I want to mention the next thing is that why jet? I mean, this is the latest technology that we developed that. So Corie pointed out that you need a concentration of hydroxyl radical. You need a steady state concentration of hydroxyl radical to modify those side chains. And now, when you come with protein, it has different kind of side chain, different kind of buffer. Maybe you have cofactors, additives. These are all like a scavenger. It will kill the OH radical. So you lose the radical if you don't put enough. So you need a very high flux. This is a flux-driven process. So your high flux density has to be very high so that whenever you expose, you have sufficient amount of hydroxyl radical present. So now, earlier we used to use the epinephrine tube, open the cap and put it in the beam. And then we started using capillary. So you have a glass and inside the solution is flowing at different speed. But we realized that these techniques have some limitations. Like if you have an epinephrine tube, you have to use a shutter, mechanical shutter. And it has a limit how fast it can open. So we have 10 milliseconds. And if you have a capillary, then you have a glass, which will also absorb the X-ray, and you lost a lot of flux density. And these days in the synchrotron, we have a mirror which can focus the beam to a tiny spot and increase the flux density quite a bit. So to use this advanced microfocus beam, we actually developed this containerless approach, like a jet, open sample. You pass the liquid jet along this microfocus beam. So we can actually use a very high flux density. And since the jet speed is very high, we can go for very tiny exposure time, like single-digit microsecond, which is very important. Why? Because we don't want to expose too long, because sample will be part up, sample will be damaged. So we want specific those tiny hydroxyl radical modifying the side chains. So that way, now the jet, we can use, as I told you, we can use regular standard 10 millimolar phosphate, 10 micromolar protein. But since we are using very high flux density, very microfocus beam, we can extend this buffer choice to tris or herpes, which earlier we couldn't do because of it was like a big scavenger, you know. So now we have slightly more choice. We can have phosphate, we can have tris, herpes, and use a very tiny and high flux density beam.
Okay, sounds good. So that is important about the low exposure time, and I'll get back to that later with another question. Okay, before that, let's switch to Lina. Lina, you're mainly involved. I should say mostly, as far as I understand, involved with the data analysis side of things, of oxidative footprinting. So I'd like to ask, like, what do you like about protein metric software when you're using data analysis?
Yeah, so I think the main thing in going from a manual analysis to the PMI software has been that in BIAS, you have one integrated platform. And it's integrated in terms of the MSMS search engine and the peptide analysis. So it's really, really nice having just one piece of software. And then I think the biggest time saver is probably that there's no need for the manual extraction of ion chromatograms, because that's definitely a very time-consuming process. And then in addition to that, you know, you get automatic peak integration. You get the calculation of percent modification is also built into BIAS. And then finally, I would say that the ability to produce customized reports is really, really helpful. So it's been a huge time saver on the data analysis side, for sure.
Great. Yeah, we're going on that time topic today. So that's great to hear. And let's get back to that actually exposure time thing. I'm curious for you know, in structural biology world, especially with destructive techniques, it's always a concern whether the technique is unfolding the protein, changing its behavior as you're doing the experiment. So so how does this, you know, you have one microsecond exposure to X-ray, but you know, how does the total destruction time, let's say, for the protein compared to typical protein unfolding rates? Anyone?
So proteins unfold, there's a range of unfolding rates depending on the complexity of the protein. I mean, I think local structures in proteins probably unfold on the order of nanoseconds, but there's, you know, larger structures that unfold on the order of hundreds of microseconds. And so the main thing I think is that OH reacts with side chains generally faster than that. So the OH reaction is happening generally faster than the unfolding. That being said, I think there's probably is some unfolding. There's some other damage that happens to the proteins during the X-ray exposure, and certainly when we're using the longer exposure time points. But what we try to do in this experiment is we try to really limit the X-ray dose to the sample. So we typically measure a dose response curve, and we look for to make sure that the change is linear as a function of X-ray exposure, which means that probably there's not a lot of other stuff happening, like the protein unfolding during the X-ray exposure. So yeah, so we try to use fast time points. We try to take advantage of the fact that hydroxyl radicals react really quickly with protein, and we try to limit the dose to limit that unfolding and other damage that's happening to the protein. Did you try to look at this possibility of any kind of other damage with mass spectrometry as well? Not a lot, no. There probably are some other reactions happening that we don't know about, that we aren't looking for specifically. Yeah. So, yeah. Okay, so for the listeners, maybe a Bionics wildcard search would be a good fit in there to try to find out any kind of unknown modification. Yeah, I mean, OH is the most reactive species to protein, for sure. Okay. All right, so I guess I get a few more questions here, and then we can call the day. So aside from on-off comparisons like binding site determination or epitope mapping, what kind of other information can you gather from X-ray footprinting mass spectrometry? Okay. Anyone? Well, I can start. So we can look at the protein interaction with other proteins. We can start looking at kinetics. So how does that happen as a function of time? How does binding happen as a function of time? Ligands and proteins in larger complexes. We can use footprinting to look at consistency of sample preparations, which might be useful for, say, the pharmaceutical industry. Mm-hmm. Can you also adjust the jet speed other than the X-ray? I guess that determines the exposure time, right? Yeah, exactly. And is that how you would control, do kinetic experiments? Yeah, exactly. We're also designing in a mixing cell, so we'll be able to, say, mix a protein with something like ATP, and then probe various time points along the process of watching some event happening with a protein. Sayan and Lena, do you want to add anything to that?
Yeah, I want to add one interesting and very exciting point about this technique or advantage. So we're using X-ray. So X-ray has some penetration depth, and it also doesn't ionize when it goes through a water like beta particle. Beta particle has high ionization efficiency. This is like a low ionizing radiation around the local environment. So it kind of penetrates a different region. So what happens if you have a bulk water in the surface, and you have a core of the protein which also has a cavity full of water? So X-ray, some of the X-ray can actually go there and actually make a local spar, a local concentration of radical. And now those radical is highly reactive. So you can actually pinpoint those core or cavity with water and label those side chains. So what has happened here, we have observed for many membrane protein, there are a lot of transmembrane helices, and some of the helices are kind of has structural water inside, like GPCR or ion transporter. So you can actually target those regions, expose the sample, and do the conformational analysis, how is open or closed form. So this is a big advantage of this technique, that it can actually detect bound water and label those residues close proximity to that. And as well as you can do now kinetics. You can mix ligand, or you can do some exchange reaction. You can do heavy water, like 18O water versus 16O water type of exchange, and find the resident time of water, which will actually give you the dynamics of the local environment inside the conformational hotspots.
That sounds super cool. All right. Okay, so I'm just looking at my list of questions here. I guess my last question would be, for again, probably for Sayan and Lena, how long does the experiment and the analysis take from like A to Z? So Corie already mentioned that you have to, submit proposals in advance, but what about the actual experiment and analysis?
Well, I mean, I can say a little bit about that, and then Sayan can probably elaborate. Assuming that you have your sample in the appropriate buffer for footprinting, the actual footprinting experiment is very fast. I mean, you could do it in, say, an hour. It obviously depends on how many samples you have to run and how many replicates you're running. And then as soon as your sample has been exposed, you would typically do like a standard overnight digest. And then at that point, your sample is more or less ready for mass spec analysis. And obviously with PMI bias as a tool, that analysis is a lot faster now than it used to be. And then in terms of the mass spec itself, that would depend on what facility you have access to. If it's in-house or you have to send out your sample. So I don't know. So Jan, you can probably talk a little bit more about the actual beamline work.
I mean, you're right about that. So I want to point out that this technique has different phases or different stages. Like you have the beamline access, which is maybe, Corie, I'm right, 15 days, like 2 weeks in advance, you have to submit a proposal. And then once everything is done, you send the sample and we discuss those things, how to make the sample. If you are in the right sample, like Alina mentioned, you get to the sample within the beamline and expose it very fast, microsecond timescale exposure. I mean, it's like 1 hour. And then you digest. And this digestion can be maybe few hour to maybe 4 hour because you got to process, sometimes you got to unfold those things, alkylate those things and clean again, for mass spec. And then the mass spec. So earlier, I just want to point out that earlier, this mass spec, like you get the data and you extract those peak area and manually assign those things. It's like, I did it myself, one peptide per day. So then it's becoming like 1 month to get a full footprint. And now we are doing like, you just get the data, put in the PMI. Lena has developed different workflow to validate those things. So I said that don't go to that detail, just give me the data, like, just run it and you can just generate the data if you have the data. And the PMI will give you in 1 hour, maybe the whole overview. And then with the overview, now you spend time to further refine those data, that maybe 7 days, maybe maximum to get it out, right? What do you think, Lena? 7 days, like,
yeah,
validate everything?
Yeah. I agree. I mean, obviously it depends on the complexity of the sample and how many replicates you're running and how many, different exposure times you're looking at. But it's definitely a fairly short process compared to what it used to be.
Sounds great, really encouraging. So I guess that concludes our discussion for today. So I don't have anything else for you. Thank you so much for joining us, all of you. And hopefully this short discussion will contribute to the understanding of your techniques in the community. Well, good luck with your work and goodbye.
Thank you.
Thank you.
Thank you.